Thank you all for coming today. We're here to have a talk on the exciting details of APS's second Nobel Prize in the last 3 years. This is a particularly gratifying one since a lot of the work was really done here at Argonne. These are really honors that the whole laboratory can be proud of. It took a lot of people to build the APS, to operate the APS, to develop the beamlines, and all of this forethought went into this great example of a piece of science that's had a big impact on society. This really takes a team effort to develop Nobel quality work like this. So the 2012 Nobel Prize in Chemistry, we were very happy to hear. It was given to Professor Robert Lefkowitz from Duke and Professor Brian Kabilka from Stanford. And Brian has been a very frequent user here at APS. They did work on the receptors on cells that allow the cells to get signals from the outside world. So it's a really important piece of work. Something like half of all medicines work on this particular type of structure on the cells. And, this is the type of work, it's easy to see why it was honored with a Nobel Prize, because it's really going to have a big impact on future medical developments. Now, none of this really would have been possible without the micro X-ray beam capabilities here at APS. And Bob Fischetti and Janet Smith are going to tell us about this. They're the ones who oversee the research and developed the techniques and the beamline that made this all possible here, the GMCA beamlines. It all started about a decade ago, I think, and has really come to fruition nicely. Janet is the scientific director, so she really sets the standard and develops the climate that allows this type of research to go on scientifically. And Bob has really driven and developed the capabilities for the micro X-ray beams, which is the technical direction that has allowed this to happen. So it's really great to have both of them here to tell us about this work. And please welcome me in joining Janet and Bob.
Thanks very much, Brian. So we don't say G-protein coupled receptor too often in biology. We call them GPCRs. And a GPCR is a protein, and it lives in the membrane of a cell. And in this low-resolution picture, it's going to be represented by a green box. And what GPCRs do is to receive signals from outside the cell, and those signals arrive in the form of molecules, usually but not always small molecules, very small molecules. And in response to getting a signal from the molecule outside the cell, the receptor directs a whole bunch of things happening inside the cell. In humans, there are at least 791 different GPCRs. And they affect physiology in 791 different ways. These drawings are not at all to scale. For those of you who aren't chemists or biologists, a protein is much larger than a little signaling molecule. In this case, the GPCR has over 6,000 atoms, whereas this signaling molecule, which is adrenaline, has about 25. So why are these things so important? Well, as Brian mentioned, about half of all drugs that are currently approved for use in the clinic somehow target GPCRs, either directly by binding to them or indirectly by affecting the availability of signaling molecules that go to the GPCRs. So I've listed some examples here. All the beta blockers block the kind of GPCR that Kabilka and his group worked on here at Argonne. Antihistamines, the asthma inhaler medicines that we use, eye drops like Visine, all those nasal sprays, caffeine, opiates, all interact with GPCRs and are signaling molecules. And some of the other drugs, the ACE inhibitors and the SSRIs that are used for treating depression, prevent or stimulate the availability of the signaling molecules to the GPCRs. So these are really, really important. So I've broken down what exactly did Bob Lefkowitz and Brian Kobilka do to earn a Nobel Prize. And I've broken this down into 4 different things. So the first thing that they discovered was that there actually is a protein receptor. So this was a really controversial idea for a long time. And the idea of a receptor was proposed over 100 years ago, but most people did not believe that most scientists who worked on signaling didn't believe there was a protein receptor until the 1970s, when Bob Lefkowitz developed the methods to detect it. And the one that he worked on is this thing that is now known as the beta-adrenergic receptor, and the signaling molecule is adrenaline. And he used a radio-labeled adrenaline to interact with it, to target the receptor and then show that there really is a protein on the surface of the cell. So the next thing that they did was to figure out which protein it was. And this was work that Brian Kobilka did when he worked in Lefkowitz's lab in the 1980s. And with the methodology that was available at the time, which was very painstaking, he isolated this adrenergic receptor from cells, purified it, and then did the really that was very difficult. And then he also determined the amino acid sequence a little bit and used that to go get the gene and determine the entire amino acid sequence of the 413 amino acid protein that is the beta-adrenergic receptor. So this led to one of the first really, really big insights in this field, and that was, it was clear from looking at the sequence of amino acids for this receptor that it was a relative of the visual receptor in our eyes, which had been studied for many, many years and was reasonably well understood. That was a huge surprise. No one expected that. And that meant that a lot of the things that had been learned about the rhodopsin system in the eye were going to be applicable to all these other GPCRs, and that was, or to the beta-adrenergic receptor. That was a huge, huge insight. The eye works differently. I'll talk about that in a minute. It's an oddball GPCR, but it is one. So then the next thing that they worked on, and they and many, many, many other laboratories, was, okay, we know there's a protein, we know what protein it is, we know the adrenaline goes and binds on the GPCR, and then what happens? Stuff happens inside the cell. Well, what is that stuff? What exactly happens? And this is where the G protein part was discovered. And I note that Al Gilman and Martin Rodbell won the 1994 Nobel Prize for discovering G proteins and what they do. So G proteins have 3 parts, which are called alpha, beta, gamma, and they usually travel together. But there are many, many other molecular players. I mean, literally, it's a cast of thousands. Many of them are signaling proteins, adenylate cyclase, calcium channels, protein kinase A, phospholipase A, it goes on and on and on. And there are also a lot of small molecules that are involved in this process that act as second messengers or carry information from one place to another, GTP, GDP, cyclic AMP, and calcium in particular. So what happens? What did all these people find out? Well, in cartoon version, when you don't really know the structure of the proteins, we draw all these blobby things. We have a cell. This is the cell's membrane, the outside side and the inside side, and the receptor sits in the membrane. And then here's this heterotrimeric G protein, alpha, beta, gamma, which is binding, that little blue thing is GDP, the small molecule. And adrenaline can bind to the receptor. Well, if adrenaline binds to the receptor, then the receptor structure is going to change in some way. If the receptor structure changes, then the G protein can bind to the receptor. If the G protein binds to the receptor, then its structure changes. And if the G protein structure changes, then it can open up and release GDP, little molecule GDP, and the little molecule GTP can bind. If GTP binds to the G protein, then alpha comes free of beta gamma, and they both go off the receptor. And if that happens, then the GTP-bound form of alpha can activate adenylate cyclase, for example. The beta gamma on their own can turn on a calcium channel that will have calcium come into the cell, for example. And eventually, when these things happen, eventually and very slowly, the G protein, the alpha part of the G protein, will convert that GTP back to GDP, so the gray molecule will go back to the blue molecule, and then this thing can reassemble. Okay. What happens, we have 791 receptors, and we have a whole bunch of G proteins as well. So depending on which tissue you're in and which receptor you are and which G alpha beta gamma is involved, many, many, many things can happen. It's not always adenylate cyclase getting activated. It's not always calcium channels. In the visual system, in your eye, we don't have adrenaline. In your eye, you have a rhodopsin GPCR with a permanently attached signaling molecule. And that signaling molecule is retinal, or carotene. It's why you have to eat carrots to see well. When light hits it, it changes its structure of the little signaling molecule inside the receptor. And then that leads to all this stuff happening. And in that case, the beta gamma stimulates the calcium into the cell, and that makes the nerve send your eye nerve go to your brain and say, you saw something. And depending on if it was a red or a green or a blue stimulated cell, you'll see red, green, or blue, or some combination thereof. In the liver, a completely different example. In the liver, this is now the beta adrenergic receptor that Kobilka worked on here. We have the ligand that comes and goes, and it's adrenaline, also known as epinephrine. This is a standard textbook picture. Every time one epinephrine or adrenaline activates one receptor in a liver cell, cyclic AMP is produced by adenyl cyclase. That activates another protein, which activates another protein, which activates another protein, which releases glucose, which ends up giving you 10,000 molecules of glucose in the bloodstream, secreted by that cell. Your liver makes a lot of glucose because you drank 6 Cokes this morning, and your insulin goes up, and then you all know what happens after that. So this particular receptor is really important for things that have to do with diabetes through that release of insulin. Okay. So the next thing that Lefkowitz and Kobilka did, well, this was the Kobilka part in particular, was to figure out how that machine works. We have this huge array of physiological responses depending on which proteins and which cell, which tissue type, et cetera. But there's a fundamental process that's the same in all of them that has to do with the receptor and the G protein. And so this is a really fundamental part of all of this unbelievably complicated signaling biology. And the only way to really understand that mechanism was to see those proteins at work in 3D, in their 3D structures. We have amino acid sequences for all of them, but we don't know what the 3D structure looks like just from the amino acid sequence. And so this has been for a very long time, a real holy grail project in structural biology. And Brian Kubilka is the one who really got it to work. There are lots of competing labs working on it, and many others have now contributed structures. But there were a lot of hurdles to overcome, technical hurdles. So the GPCR is in the membrane of the cell. That makes it very challenging to isolate from the cell and purify in a state that doesn't have it completely beaten up and non-functional. The GPCR is also wobbly because remember the structure changes depending on whether you have adrenaline bound or the G protein bound. Everybody's structure changes a little bit. And it was necessary to find a form that could be crystallized. So there were several receptor structures, the receptor part alone, and then there were the real result that put this completely over the top for the Nobel Prize was a structure that had the receptor and the G protein. And that work, that took a village to do. This is a really, really tough problem. So here's this blown up cartoon of the G protein binding to the receptor, but it required some very clever protein engineering, adding a viral protein to help it crystallize. This was developed in Kubilka's lab. There was an antibody added from a llama. That came from a lab of Steyert in Brussels. There were some special biochemical tools developed to trap this. You remember this, we have this open and close business to release the GDP. Roger Sunohara at Michigan had the biochemical tools to do that. He was involved in this project. And then there was electron microscopy to visualize this complex at super duper low resolution by Yorgos Skiniotis, also at Michigan, to screen all these samples and figure out which ones actually had trapped a single form that could be crystallized. And then there were some new crystallization methods from Martin Caffrey's lab and reagents from Martin Caffrey's lab at Trinity College in Dublin. And there was a lot of expertise in crystallography from Bill Weiss at Stanford. And that's what led to the structures. And now I am going to turn it over to Bob to tell you a little bit about the structure determination.
I don't need that one. Okay. Can we switch microphones? Yes. It's switched? Great. Okay. All right, so we'll switch a little gears now and talk about some of the developments that have been done here that help to enable this science. So first off, Janet mentioned that we need 3D structures. So how do we get the 3D structure? So we start out with a small crystal. With the X-ray beams that we have, we get a diffraction pattern. From that, we get an electron density map, which you may not be able to see very well, sorry. And then from that, we get the atomic structure of the molecule. And that's basically what this is all about in a nutshell, but it's obviously a lot more complicated than that. One of the biggest issues is that protein crystals are made of low Z elements, so they scatter very weakly. They also have a lot of solvent and water in them. So again, they scatter very weakly. So we need a strong source to be able to get the intensity out of these weakly scattering crystals. Conventional sources, home sources, took a long time to collect data. You had to worry about stability issues. And so it was very difficult to get high resolution data in the early days. Synchrotron radiation has had a tremendous impact. The high intensity, the high collimation of the beam allows us to work with smaller crystals and to get the signal out of these small, weakly scattering samples. And we use techniques like cryocooling the sample to try and help deal with issues like radiation damage, to have the sample last longer so that you can collect data to high resolution. But the bottom line is that the high intensity beams that we get on the undulator beams at synchrotrons, you get better signal to noise, and that's what it's all about. So I want to go quickly, a brief tour of X-rays and structural biology. This is a very dense slide, so bear with me. It all started back in 1985 with Röntgen's discovery of X-rays. And one of the first exposures he took was of his wife's hand, showing the bones and her ring. A few years later, von Laue discovered the diffraction of X-rays. And not far behind that, the Braggs working together, father-son pair, to determine the crystal structure of the first crystals. It wasn't until 1947 that synchrotron radiation was actually observed. So it's kind of interesting. That's not a Nobel Prize, but it was the first observation of it. The next Nobel Prize related to this was the first protein sequence that was discovered by Sanger for insulin, followed by Watson, Crick, and Wilson's determining the structure of DNA. And then they used Rosalind Franklin's diffraction pattern, and they got input from Jerry Donahue, who helped figure out the base pairing, how the DNA would form together. I had the privilege to know Jerry. He was chair of my thesis advisory committee at the University of Pennsylvania. Right behind that, this actually the same year, was the award to Kendrew and Perutz for the first 3D structures of myoglobin and hemoglobin. And this is a picture of the model that Max Perutz made of hemoglobin that I took from Cambridge University. It's on a ledge that you can see. And here's an experiment back in 1984 where I got to work with Max Perutz doing some X-ray spectroscopy at SSRL. In 1964, Dorothy Hodgson got the structures of penicillin and vitamin B12. And then the first diffraction pattern using synchrotron radiation was actually from Gerd Rosenbaum at Daisy in 1971. I think Gerd's probably here. I don't see him. Following, not far behind, was the first diffraction pattern from a crystal using synchrotron radiation from Keith Hodgson's group at SSRL in 1976. And then Aaron Klug determined the 3D structure of a nucleic acid protein complex, followed by Deisenhofer, Huber, and Michels determining the first membrane protein structure. And then, as Janet mentioned, Gilman and Riddell got the Nobel Prize for the discovery of G proteins, and then Boyer and Walker for understanding the mechanism and the structure of the ATPase synthase. And then more recently, McKinnon got the Nobel Prize for solving the structure of a very important integral membrane protein, potassium ion channel pump. And then Kornberg for the structure of RNA polymerase. And then, as we all in this room probably know, the 2009 Nobel Prize in Chemistry to Ramakrishnan, Straits, and Yonath for the ribosome structure. And some of that work was done at SBC here at the APS. And then finally, the 2012 Prize in Chemistry to Kobylka and Lefkowitz for the G protein coupled receptor. So there's been a lot of activity over the last 100 years, and this is just sort of a brief snapshot of some of the things that I thought were relevant to getting to the end results here that are of interest to us. I'm not sure I need this slide for this crowd. I wasn't sure how broad an audience we'd have, but just very, very briefly, if you have an accelerated particle, it'll give you dipole radiation, sort of a globular pattern. And if you accelerate that particle to relativistic energies, then you get this nice forward focus. And if you have the advantage of working at a facility that has an undulator source, which causes the beam to oscillate and gives you coherent interactions, you then get a very narrowly focused, coherent or partially coherent beam that has particular harmonics. And it's a very intense, very well collimated beam. And just a cartoon of a synchrotron, then beam goes around the loop. When it comes to a wiggler and oscillates, it emits this laser-like synchrotron radiation, and when it goes through a bending magnet, it emits the lower intensity, more continuous spectrum bending magnet radiation. So just to give a feel for where have we come over the years with the intensity of sources and how have they impacted structural biology. In the 60s, most of the work was being done with rotating anodes, and you had intensities, this is sort of 10 to the 10. This is a logarithmic scale, and it's in brilliance, which now the more proper term is brightness, but this is an old plot. In the 70s, bending magnet sources became very common, and it was sort of maybe more like, you might say, a flashlight instead of a light bulb. And then finally, in the 80s, we got access to undulator sources, and this really gave us this laser pointer-like intensities that have enabled some of the exciting sciences that are going on these days. And this is sort of the region of interest for most of us in here, in the sort of a few kilovolts to 10, 20, 30 kilovolts. And so these are the various intensities that one can get at different sources. And one of the things that's interesting to see is since the first structure was solved, of a protein or a DNA in 53 and then myoglobin in 57. There's sort of been a trickle of structures being solved as time went on. And this cartoons down here just sort of show when the sources in the US came online. But what's really interesting is to see the recent uptick in the number of structures that have been solved and whose coordinates, atomic coordinates, have been deposited into the protein data bank. And so what I'd like to do is to try to explain a little bit about what's given rise to this large increase in the rate of structures being solved. Well, here at the APS, we have 8 sectors that support macromolecular crystallography. And we have contributed about 20% of all of the structures that are in this database. So it's a really significant achievement. But what's also important is not just the beamlines, but the facility, the source itself. And one of the things that has been amazing is things like top-up, the stability that we've gained from having top-up that was developed here at the APS. And just having a beam that you know is going to be there all the time, having 98% availability is incredible. And no synchrotron has in the past worked like that. Just having the availability, when you turn the switch on, it's there. Makes all the difference in the world and in the science that you can do. So what are some of the technological advancements that have led to this rise in the number of structures being solved? Well, certainly it's the availability of undulator beamlines with high intensity, the parallel beam, which allows us to look at maybe larger unit cells or structures of complexes like the GPCRs, and just the stability makes a huge difference. Also important have been improvements in detector technologies. We're not using film anymore. We now have electronic detectors, either charge-coupled devices or pixel array detectors. And there's also been advancements in the biology, advances in molecular biology and protein engineering, but also improvements in crystallization. And we're looking forward to the new building that's being constructed, the Advanced Protein Crystallization Facility, which will also help to facilitate that. Also important was the development of cryocooling to minimize the effects of radiation damage. And one thing that maybe doesn't get stressed as often is the automation of all the processes, the automation of mounting the sample, the automation of centering it, the automation of collecting the data. It's made a tremendous difference in what people have been able to do. Another aspect has just been computational in terms of having the availability of fast workstations and being able to, in real time, see a display of a molecule and see if it's really, have you solved the structure. And it's made a tremendous difference. Another step forward has been the development of microcrystallography. Working with small crystals means that we need small beams. And some of the advantages of working with small beams is that you can reduce the background, you can get improved signal to noise, and it's all, as I said earlier, it's all about signal to noise, trying to get the data, good data, out of the crystal. It also helps with larger crystals, not just small crystals. Crystals are not always homogeneous. You may have a big crystal, but it may have a lot of mosaic crystallites within it. If you can use a small enough beam to locate that particular well-ordered crystallite, then you can get good diffraction from an otherwise crappy crystal. That's been a huge advantage. Another aspect of small crystals is they're clearly easier to grow. It's hard to grow a big crystal often. They're more successfully cryocooled. They don't tend to be as disordered by the cryocooling process. And often, you just get a crystal that has better ordering than the small crystals. So at GMCA, we have 2 beamlines, 2 insertion device beamlines. They're canted undulator beamlines. Many of you in this room probably have already seen this slide. Some of the aspects that I want to talk about is that what have been important is the ability to rapidly tune the intensity and to have both high intensity and positional instability for these experiments. And part of what we have done at GMCA is we use bimorph mirrors, which have allowed us to get a small homogeneous beam. But in addition, we've developed what we call mini beams using our mini beam collimators. And that's what really has changed the way things are being done and has enabled the science that Kobylka and his group have been doing. And we have these collimators on both beamlines, and I'll show you some of that. We've also done some work with zone plates, but that's not relevant for this talk. So in early 2007, we were developing the small beam capability. And so in February 2007, we had this device which had just a single beam collimator. Here's the sample on the goniometer, and here's a lens that is actually looking at the sample. The beam comes through the lens. So it's a what you see is what you get type setup. It was about this same time Kobylka heard that we were developing this and asked for beam time. And so he came and started using this beam. Now, as you might imagine, when you first develop something, it's not the most stable, not the most robust. And from pressure from Kobilka to, they had key science that needed to be done. We kept developing, working over the years. We went to a triple, a more robust triple, and then finally a quad beam collimator, which allows us to have a 5, 10, or 20 micron diameter beam, or a larger scatter guard for the full beam. And that's really changed the way that users can do science. And this was driven by work like Brian Kobilka's. So one of the first experiments that they did here in solving the structure of a GPCR, they had crystals that looked like this. And if you used a large beam like this, the full beam, you'd be illuminating not only the crystal, but a lot of mother liquor around it, and you'd get a lot of background. By choosing a smaller part of the beam, you could make it a smaller beam and illuminating only crystal, you got enhanced signal to noise, and they were able to mitigate the effects of radiation damage by collecting partial data sets at each of these spots and merging that to get a full data set. And that resulted in the first paper in Nature in 2007, and Nukri Shanasvili and myself were co-authors on that paper with them. And again, it relied on our mini beam technology. Another step that Janet mentioned was the introduction of a new way of crystallizing the proteins in the so-called lipidic cubic phase, which came from Martin Caffrey's group. Vadim Cherezov brought that over to Ray Stevens' group, and they collaborated with Kobilka, and were able to crystallize the GPCR into a much higher or much more well-ordered structure that could then give a much higher resolution pattern. This is what a drop of crystals in the lipidic cubic phase look like. It's a shower of tiny, tiny little crystals. Problem is when you put it in a loop and put it on the beamline, the dots here show where the little crystallites are. You can't see them. The lipidic cubic phase becomes opaque when it's cryocooled, so you have a problem. You have crystals, but you don't know where they are. You need to have a way to find them. So I'll come up to that in a minute as to how we discover them, but one of the things that we developed along the way to help users, in particular groups like Kobilka, was so we start out with the beam on, say, a YAG crystal so we can visualize it at the sample position. But the user has the ability to just select a 20 micron beam, a 10 micron beam, or a 5 micron beam. And it literally takes about that long. That gives them the ability to start out, if they've got a large crystal, start out with a large beam. If the diffraction looks good, collect data. If it looks poor, then you go to a smaller beam and you start trying to find the homogeneous regions of the crystal. Or if you have a small crystal, you want to use a small beam. You get the best signal to noise by matching the beam size to the crystal size. There's several other aspects that were important for microcrystallography that we were developing over the years. If you're going to have small crystals that maybe they're not in the lipidic cubic phase, but you can see them, then you want good optics so that you can visually see it and align it. You need the mini beam collimator so that you can match the beam size. If you have a small crystal and a small beam, and you want to collect data, you need to be able to rotate that crystal to collect a diffraction pattern over a wide range of angles, but keep the crystal in the beam. So having a small sphere of confusion goniometer is important. You also want to know that the intensity that you're measuring is stable and things like that, so we have an active beam stop. And we also had done some experiments using nano positioning for radiation damage experiments. So it's not just the hardware. A key component of what we developed in our group was the software to combine the two to make a powerful tool. So one of the things that we worked on was so-called rastering. This sort of has its roots back from ID13 at the SRF, where they call it mesh scans. And then there was an implementation at SSRL in 2007. And we took that concept and expanded and just have been running with it ever since. And it's become a very useful technique for users, and just about every user says that they love these new tools that we've developed on the beamlines. Basically what it is if you have a crystal, you could draw a box, and you could go and step along and take diffraction patterns at various locations and get a sort of a heat map that tells you how well that diffracts. Or if there was several small crystals, you would locate the small crystals. Well, if you could see the crystal, you don't need to do that. The problem is, when you have a sample in lipidic cubic phase, it looks like that. You have no idea where the crystal is. So that's where these tools really come in handy. Another aspect is automating the entire process. So now we can, on the beamline, the sample automounter will take up a sample, put it on the goniometer. It'll determine the profile of the loop that's holding the crystal, center the loop. It'll then transfer those coordinates automatically to the diffractive rastering routines, which can automatically now collect the patterns and tell you where your crystal is and hand that over then to the data collection routines. So more and more of the processes are being automated for users so they can spend more time looking at the quality of their data rather than trying to look at how do I get the data on the beamline. Another aspect is some samples most have some radiation sensitivity, others more, some more than others. And one aspect of tool that we've developed is so-called vector collect, where you can collect along a three-dimensional vector. You can either collect partial data sets to be merged into a full data set, mitigating the effects of radiation damage that way, or you can do a continuous screw data collection to just try to make the most use of all of the protein that's there in the crystal. So in the last several years, Kobilka's group has solved several structures related to the beta-adrenergic receptor that led to the final complex structure. So there's these 7 or so structures. The first one was based on work that was done initially at the SRF and then here at the APS, the work in 2007, and then all the subsequent work was done here at the APS. And this is the work that really was the holy grail that led to the Nobel Prize. This is an image that was captured off of the video from Brian Kobilka's Nobel seminar. And this is a picture of them. This is Brian, and this is his collaborator, Roger, several other group members that have worked with them. This is a picture as they have just finished collecting the data on the complex structure, and they're heading to O'Hare. And Brian's holding the camera and taking a picture of everyone in the group. And they also acknowledged the work that the work was done here at the APS and at GMCA. And these were images of the final crystals that they were able to get for the complex. There are now many groups working on GPCRs, and mostly I would say it's been because of the success of Kobilka's group. Others are applying this technology, and there's quite a bit of activity in the area. So Brian's group, for example, has 12 structures of 6 different GPCRs. Ray Stevens' groups and several others are also all working on structures. This was early work that was done on rhodopsin that Janet mentioned, that was done back in 2000 at SBC. We also have several proprietary groups who are working on GPCRs with a particular interest in finding particular molecules that they can bring to market. So there's a lot of interest in this area. In terms of publications on GPCRs, there's now 23 publications from GMCA. There's the one, but there may have been more. I'm sorry if I'm not sure on the rhodopsin structure from SBC. And these include the M2 and M3 muscarinic acetylcholine receptors, the 3 opiate receptors and the opiate-like receptor, as I mentioned, rhodopsin. And in competition, there are 6 publications so far from other light sources, from the Diamond Light Source and from the SRF. And at this point, I will switch back to Janet.
Okay, thanks, Bob. Is my microphone back on? Okay, good. So after all that technology and all those, we counted something like 45 days of beam time between 2007, when Kobilka's group first came to GMCA, and 2000, and this fall when the prize was announced. It was, I have to say, really heartening to see the enthusiasm that these guys had, and coming back again, and every time it would get a little bit better. And some trips were not so good and some were better. But the last one with the photo in the car was really, they knew they had it. They knew they had the big one. So at that point, although they hadn't quite solved it yet. But what did they find? Okay, so I'm going to come a little bit back to the biology. So this is the structure of the complex, the signaling complex, with the adrenergic receptor in the membrane, or in a detergent micelle in the crystals, and then this heterotrimeric G alpha beta gamma signaling molecule bound to it, and here's a perpendicular view of the same thing. So to remind you of what I said earlier, this is the receptor changes structure when adrenaline binds. So here's where the adrenaline binds up here. And the green is the structure, is what the receptor looks like when it's active, when the signaling molecule has bound, and the blue is what it's like when it isn't active. And the structure changes, not in any huge, enormous way, but in this, see this little shift of a green helical structure here, the blue helical structure shifts over. Doesn't look like much in this orientation, but if you look from the bottom, you can see that it shifted 14 angstroms. That's a long way. Well, that's the structure change that lets the G protein bind. And the thing that moves is this transmembrane, trans across the membrane, helix number 6, shifts out of the way. Here in magenta is the unactivated way. It gets activated, boom, and that's exactly where the G protein binds into the receptor. And they have a lot of details in the paper that I didn't show here about how binding adrenaline here leads to tiny little changes that result in that larger helical shift. Well, then what else happens? Well, remember the G alpha, the G protein, also changes its structure when it binds to the receptor. And that you can see, here's the structure they determined, the green and the yellow. Beta gamma are left out so you can see in this picture. And there are many G protein 3D structures now in many different states. So we understand how nucleotides, the GDP, GTP, bind to the G protein. And the gray structure is one of those other G protein alone results that had been published some time ago. And here's the GTP bound inside, and this is when it's closed. And this mouth opens, and one of the other surprises of the big complex structure that Brian Kabilka's group did is that it opens all the way up there. It's a huge opening, much larger than anybody expected. And that was a major, major surprise, which I have to say the field is still digesting. The implications of this are not in yet. And there are a lot more structural details in their publications, and this now gives us a big picture. And the details that are in here will explain how a particular G protein might interact with one receptor but not with another receptor, and many, many other questions like that. Remember we said Brian's Kabilka's group had done 12 different 3D structures of 6 different receptors. Well, 6 of those structures are of the beta adrenergic, or 7 of those structures are of the beta adrenergic receptor, this very green molecule that's here, but they have captured it not just with a G protein, but with a signaling molecule that turns it on, and with a signaling molecule that turns it off, and with a drug that blocks its activity. And all the tiny little details that are in there are the things that are going to be so important for where this is all going that will matter to everybody. So looking forward, we're going to have we now have a much better understanding of this generic mechanism for signaling of 791 receptors through I don't know how many different G proteins. We're going to understand, we already understand much better how the drugs work that interact with those receptors. We should be able to develop improved drugs that are better targeted for the specific GPCR they should interact with and not with others. And we also might be able to develop new drugs that, rather than being targeted to where that adrenaline goes, possibly could block some of the protein-protein interfaces that exist in these complex structures that no one had seen before. And in addition, there are several improvements that we expect in the area of the X-rays, and Bob will finish up with that.
That includes developing a new microfocus beamline to give us smaller, hotter beams for more challenging problems like these complexes that they're studying. Faster, higher efficiency X-ray detectors are becoming available, and in fact, we have one on order. and more intense and stable X-ray beams that we expect to get out of the APS upgrade. And looking really to the future, the X-ray free electron laser, I think the jury is still out as to what advantages that'll have for this type of science. There's certainly a lot of excitement, but there's also some significant challenges that need to be overcome before it replaces synchrotrons. And then finally, this uptick is going to continue to rise. We believe, this is from a paper that Janet Smith and I and Masaki Yamamoto at Spring 8 wrote, summarizing microcrystallography. And there are already now about 24 beamlines that have beams smaller than 20 microns, and there's another 10 or 15 beamlines either in development or being commissioned. So there's just going to be this continuous explosion in the availability of high quality small beams for projects like these. And so it's going to really have a huge impact. And then finally, we could not have done this without the entire GMCA group. We've been fortunate to have a very talented staff of hardware developers, programmers, crystallographers, and what's really key is they all work together to develop these tools, to make the hardware and the software sing together, and to have Kobilka smile and take a picture as he heads to the airport. Thank you.